Okay, so for the third part of this topic, okay, because we are on the separation based on electric forces, so we will continue with the 2D forces, which is on two-dimensional. Sometimes you say 2D, sometimes you say 2DE, they are the same thing. So 2D separates protein in two independent properties into distinct steps. So that's why in the first dimension, it is the application of the IEF or isoelectric focusing. And what happens in IEF is that proteins are separated according to their isoelectric point or pi. I'm sure you guys have heard of pi. Dr. Hayati would have covered it in her protein separation. And in the second dimension, after your protein have been separated based on the pi, so it is basically SDS page whereby you separate it now based on the size. Okay, so for the separation. Alright, so this is a representation of IF. This is taken from my PhD thesis. So, in the first dimension, okay, you have all proteins mixed together in a mixture. So before focusing, you have different proteins at different pi. So after focusing, okay, what happens is that okay, you now have pulled up okay your protein okay according to the pi. So this is okay a mixture of protein before IF and how they are then separated based on their pi after focusing. So this is done okay on a strip. Okay, so a plastic strip okay but with a gel on top of it okay, and your protein is loaded okay, and then when you apply a certain voltage at certain time okay and gradiently or and not okay, so it's actually a very complex procedure. Then after a certain period of time okay, usually around 5 up until almost 24 hours or more than that depending on your protocol, you can get it separated after focusing based on the pi. So well, if you remember back okay, pi and pH, what are the correlations? So well, I will ask that okay when we meet okay online, but okay think about that, find the answer. So after IF, what happens is that okay your pi equals to pH. All right, now so what happens again over here okay before okay or before you run it okay into 2D okay on the two dimensions, so everything has gone through the one dimensional IEF okay, so you now have separated based on the pi, so now okay it's equivalent to the pH. So equilibrate in SDS plus reducing agent to give the uniform shape single subunit blah 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 and so forth. Okay, so there are two types okay of reducing agent, which is the DDT and also the iodoestamide, and then you basically okay, make sure okay, that you run it on the gel. So running of the protein gels in 2D is similar to the 1D. It's just that most of the time we don't have the stacking gels. Okay, we just have one gel which is just resolving because you don't really need to make wells. Okay, over there on top that you only have one well, or usually we don't have any well at all because we just put the strip unless you want to have one well for the marker just for guidance of the sizes. So similarly with the basic application of electrophoresis, after okay, running it for a certain period of time okay, so negative to positive, so you can now have the protein which has firstly okay, in the first dimension separated based on the pi, it is now separated further based on the size. Now so this is more or less like a rough representation of how 2D is being done from your 1D over here. Now it is run 2D. So your protein basically moves according to size. Okay, so if you want to have a look again, okay, so this is how the 1D occurs. Okay, it is now separated based on the pi and pH. So pi now equals to pH in 2D. Okay, so your protein will now travel okay or separated further based on size. So over here you have okay, a very specific size with a certain pH. So the range of pH basically depends on what you want to use. Okay, so 3 to 10 is most common. You can have a bit acidic or a bit basic depending on your protein or your targeted protein. Alright, so this is a rough estimation of the 2D page okay, of a lung V79 cells from CHO okay, or Chinese hamster okay, which is later on okay, used for cancer cells. You can see that again there are lots of dots over here or spots as we refer to them okay, as compared to bands in 1D. So what are the differences? I think I mentioned it previously okay, in the first week okay, so try to remember back okay, because okay, we will discuss them, okay, when we meet online later on. Now, for the 2D, so it produces protein spots where each spot represents a single protein species in the sample. So this would enable separation of huge amount of different proteins with information on the protein PI, the molecular weight, and also the amount of each protein, a rough estimation at least. So this is the general flow of proteomics work, okay, and especially if it involves 2D. So from your protein samples, okay, you basically separate it based on 2D. You get all the spots, you excise a spot or more than that. You digest it with trypsin, one of the protease, the most commonly used. And this is especially for protein profiling. So what happens is that, okay, you either go through multi or you go through the LCMS, okay, and then, okay, you get your peptide sequences, okay. So it's actually both processes are called PMF, not PMS, okay, peptide mass fingerprint. Okay, you get your protein sequences, sorry, peptide sequences, and then you could basically could come up with your protein sequence just doing a blast search. Okay, so further on 2D SDS pH gel. So in the first dimension, okay, so to recap, okay, it is separated by isoelectric focusing. So the gels, okay, this is on a strip of plastic, okay, there is a gel, okay, immobilized based on the pH gradient. For example, 3 to 10, like I was saying, okay, this is the most common. So then you put it, okay, in a machine, well, a very specific machine, okay, called the IEF, okay, isoelectric focusing machine. So electric current, okay, will cause charged proteins to move until it reaches the isoelectric point, your PI. So pH gradient makes the net charge 0, which means that pH is now equals to your PI. Then later on, okay, you can just separate on your SDS page, okay, which is the 2-dimensional, and then get your spot and you can further analyze the spots. So this is one of the examples, okay, of the IEF system. So there are many different types of system depending on the brand. So this is from GE Healthcare. So it accommodates all length of immobile dry strip gels. So the strip of the gels, there are in variable size, okay. So 7, 11, 13, 18, and the maximum is 24 cm. So that means that you can have as little as a small gel, okay, which is the most common, 7 cm, okay, up until the 24 cm gels, which is the largest, especially it would be useful if you're doing GIH. Okay? And of course, okay, it could run up until 12 gels simultaneously. And because protein work has to be reproducible, usually we run at least 2 strips at one go. Usually it's recommended to 3, but of course this is actually very expensive, so usually we just run 2. So this is the process, okay, for first dimension, okay. So a rehydration solution is actually applied to the strip, okay, because the strip actually there is dehydrated gels on it, so the gel has to be rehydrated. So you apply rehydration solution, so it has to have a specific mixture, okay, for your protein for IEF later on. So what happens, okay, is that, okay, when you put it, okay, in, well, you can use a very specific kind of strip holder over here, or you can just use whatever holder that you can get your hands on, but make sure that it could fit your strip. Okay, so usually apart from the single strip holder, they have a 12 strip holder which is actually made in plastic, or you can have it in glass. So first and foremost, once you have put in the rehydration solution, usually we put in the rehydration solution first, and then you remove, okay, the protective film from the dry strip gel. So this is the film that you need to remove, okay. Your gel is on top, okay. So the gel is going to be kind of like sticky, so try not to touch it, okay, with your hands, even though you are wearing gloves. So the dry strip, okay, is put in the strip holder with your rehydration solution on it, so gel side down. Okay, so you need to remember to flip it the other way around. If you were to put the plastic here, so your gel will not be rehydrated. Okay, so afterwards we apply cover oil. This is to ensure that rehydration occurs smoothly. So cover oil would be in forms of mineral oil, or basically is a bit better as compared to mineral oil, so it's a bit pricey actually. And then you cover, okay, the strip holder, okay. So over here, like I said, this is very fancy. Usually I just cover it using the aluminum foil, that's it, okay. And then leave it to rehydrate overnight, so in a dark condition, of course, since you also have covered it, okay. Or you can put it, okay, in the machine if you want to further enhance the separation technique. So you can do a passive, okay. What I meant by passive is that you just put it in room temperature, okay, leave it overnight, okay. Or you can just, okay, put it, okay, in the IEF machine, okay, put some or use some procedure, which means apply some current, okay, for the overnight procedure, okay. That is the active rehydration. It could also be done. So these are some of the links to the videos if you're interested. So usually, even if a physical class, I don't show all of them because it's actually quite lengthy, and it is actually an interesting procedure, but a tedious at that. So usually it will take around 2 to 3 days to run to the page. Now let's have a look, okay, at the results. Okay, so let's take a look at some fancy picture of the protein spots. So we have two gels here, your control and your experimental gels. So of course, okay, I'm not showing you the spots, okay, because they have been covered by numbers. So these are how many spots that they counted. So you can see up here 1, 2, 6, 5, okay, more than 1,000 spots again have been counted. And you can count either by your naked eye or you can use a spot cutting machine, okay, which could count for you with the software installed in it. So now let's have a look, okay, at these two gels, okay, furthermore. So basically without the numbering. Now focus on the ones, okay, that has been more or less circled, okay. So this is of mouse cardiac, okay, same amount, okay, 250 microgram, okay, same pH strip, okay, pH 3 to pH 10, okay, same concentration of gels, 12.5%, okay. But okay, there are differences in the control and also in the experiment, okay. So what do you think occur here? Okay, is this the same as this? And what happened to this? Because it's not obvious in here, but you have something over here. What happened? Think about it. I may ask in the online class. Okay, so at another point of the same gel, okay, so this is just a blow up, okay, or a close up of the control versus the experiment. Okay, so actually this is the same one, okay, just now, okay, so that you can actually have a clearer picture of it. Okay, so this is the one similar, even though similar, but you can see that this is kind of like nice circle to it. Okay, this is kind of like a big circle, kind of like a bit oblong, isn't it? Okay? And of course this is the one that we see which is different. Okay? What are they? What happened? There are a few deductions that you can make. Alright, so this is another one. Okay, so this is the other one, okay, from the same gel but different location. Control versus experimental. Okay, so we can see that, okay, these two are the same, so we know that these two are the same. But then, okay, in your control, the spot is here. In your experiment, okay, it's not here, it's here. So what happens? Okay, so there are a few postulation or deduction that could be made. Okay, they made over here. Either is it PTM, post translation modification, or is it down regulation, okay, or up regulation? Yeah, so think about that. There are a few deductions that you can make. So the last one I'm going to cover is basically on DIGE, okay, or differential gel electrophoresis. So usually when you do a comparative proteomics, okay, comparative proteomes, so they are labeled prior to running the first dimension, whereby you can have up until 3 proteomes, okay, and using 3 different fluorescent dyes. Usually it is not recommended to have more than 3, and usually people just run 2 because even for 3 it's actually quite hard, okay, to see the color sometimes, especially if they overlap. So what happens is that, okay, you mix the label standard, okay, so you label it separately but then you mix it up together, okay, and then, okay, you run it on one gel only, not on 3 separate gels, okay, only on one gel. Okay, and of course you need to have an internal standard, okay, to make sure that you don't lose track of the spots later on. So allows use of an internal standard in each gel to gel variation, reduces the number of gels, okay, to be run. And okay, even though you only get one gel, okay, because this is dyed, okay, or basically labeled, okay, with fluorescent dyes, okay, you can actually have 3 different gels, okay, using different excitation wavelength corresponding to different dye. So of course this has to be done, okay, using the software, okay. So what you could deduce, okay, from over there, okay, is that this is whatever that you see, okay, basically based on the mixture, but okay, you can have it separately, okay, and then you can actually just analyze the image. This is very useful if you wanted to see if there is an upregulation or downregulation or PTM isomers, okay, being created and whatnot. Okay, so that is actually very useful. Not easy to do and not very cost effective, okay, but a very good experiment of comparative proteomics nonetheless. Alright, so let's continue on to get towards the last part, which is on protein visualization on the gels. So we have covered this last week. Okay? So protein visualization on gels is similar over here. So usually after electrophoresis, the gels are precipitated by either adding alcohol containing solutions or strong acids. Okay, for example TCA, and the proteins are often stained by either Kumasi blue, okay, or by silver staining. And apart from that, there are also many other stains which we have covered. Okay, so there are about 5 stains we covered from last week. So these are the differences, okay, and you have seen it, okay, previously as well. So I'm actually just only showing you, okay, the 1D because I did the 2D but only with Kumasi but not with Civil State, so I don't have the comparison. So you can see, okay, this is from my PhD work, okay. So they are the same proteome, okay, from Arabidopsis phloem, okay, but as you can see, okay, how they differ from when they are stained using Kumasi as compared to when they are silver stained. Okay, so they are the same. It's just that probably you can see the bands better over here as compared to here. Some of them at least. Okay, so there you have it. Okay, so this is about the separation based on the process. So next week we'll continue on with separation based on chromatography. So before we move on, I would just like to say that you can actually, most of the time it depends on the researcher preference. Either you do the separation on electrophoresis first, okay, or you proceed with the purification or separation by chromatography then with electrophoresis. So there are two ways of doing it.